Good afternoon, my name is Jefferson Susoni, and today I want to talk to you about a problem our society faces. And that problem is that of alcohol. CDC estimates that 88,000 people perish to alcohol consumption every year. Unfortunately, our society has deemed alcohol as the lubricant of choice for social interaction. This places people with a natural susceptibility to alcohol in a precarious situation in which they have to engage with social norms, and because of their natural sensitivity, they are more prompt to developing issues with alcohol, especially with alcohol consumption. The mechanisms that confer these inclinations remain elusive, while the question remains, are there genes that impact alcohol sensitivity or resistance? A research published in the Most Psychology Journal from the Department of Molecular Biology in the University of Texas Southwestern Medical Center set out to find SNPs which aid in the desire for consumption of alcohol. As we know, flies seek for items that have high contents of sugar in their natural environment. As these items decompose, small amounts of ethanol are produced naturally, and when the flies go attempt to ingest this, they inevitably sometimes become inebriated. Now, that sets a response in which they seek to find items that the smell of alcohol, because it indicates that there is sugar content, and because of that they follow through with consuming the alcohol. Now, the interesting thing is that some genetic makeups increase or decrease the sensitivity a fly has to alcohol, just like humans do. We do not know what those specific genes are, and that is what we are trying to find. So, the Department of Molecular Biology, in their search, they found potential stocks which show a natural desire for alcohol prior to being exposed for the first time, meaning that they seek to consume alcohol before having ingested. That is really interesting. And another notable thing is that they displayed a higher sensitivity to alcohol. And how did that happen? They created these mutants by affecting their GT phase ARF6, which is required for normal ethanol-induced sedation in adult fruit flies. Now, what does that do? ARF6 was first implicated in phagocytosis in 1998, and it is part of the cascade of events prior to phagocytosis. It is also implicated in the regulation of ethanol-induced behaviors. The RHO family GT-PACE RAC1 acts in conjunction with the ARF6 to regulate ethanol-induced behaviors, like I stated earlier. Both ARF6 and AEFA6 are required for ethanol-induced tolerance, a behavioral change upon repeat alcohol exposure that is thought to be a precursor to alcohol addiction. And why is this important? Because the team set out to look at knockout strains that were mutant and displayed a higher tendency to be sensitive to the alcohol, meaning that it took less time for them to become inebriated as opposed to the wild type. Here we're going to explain. The x-axis represents the ST50, the y-axis represents the fly type, and this is the wild type right over here on the top. It is the gray area, the gray bar. The three mutants that were studied are the ARFIP, the ARF6, EFA. And these stocks of mutant flies display a higher sensitivity and innate desire to consume alcohol prior to being exposed. Dr. Keys and the team at NCCU set out to answer this question. We used Drosophila melanogaster as the organism to observe. We used this organism because it shares 60% genes homologs with humans, thus making the fly a perfect organism for observations. We tapped into the living library of organisms named DGRP. DGRP stands for Drosophila Genetics Reference Panel. Out of that panel, we took stocks, and these stocks were unique because of the SNPs that they contain in their genome. Now, SNP stands for single nucleotide polymorphisms. So our methods. We ran 3 to 5 sets of observations in triplicate, and the following stocks were 324, 859, 303, 714, 712, and 99. We compared these stocks to a wild type, which was kept the same throughout all of the observations. Flies were also separated by sex and were secluded in vials of 7 to 10 flies per vial. These vials were observed for ST50. ST50 stands for 50% of the amount of flies in the vial to become sedated. ST stands for sedated time. On to the data. And the data is set out in this Excel sheet. Each observation was done, and we have 2 books, one for the male, one for the female. We're going to start with the male. So if we look at these highlighted portions, this is the time where ST50 was achieved. ST50 stands for sedated time, and 50 is the 50% of the amount of flies inside the vial. Now if we look here at the upper left, we can see how the data was arranged. The temperature was at a constant 22 Celsius. The next line, we have the type of the fly, the date, and the time of the observation. On the next line, we have again the type plus the number of flies inside each vial. And like I said, this is the time, the y-axis, right? This is the time, and this is the highlighted ST50. If we go to the right, we can see the actual calculations, and we're going to zoom out slightly here. Perfect. Okay, so here we can see the calculations. We can also see the t-test and the standard deviation. And now we're going to explain the graph bar of the male observations. The stocks that we observed are the W1118, 303, 859, 324, 714, 712, and 799. The y-axis displays the time that it took for the flies to succumb to the alcohol. The x-axis, of course, stands for the stocks that were used. We see the error bars here, and they overlap. Some of them do not. The ones that overlap indicate that we can be certain of the data and their error margins, and that they're close together. And the ones that do not overlap, we cannot be certain of that, likely because we did not have enough observations. You see, 714, 712, 799 did not have as many observations as 324, which was the one that we observed the most. Another important thing here is that we did the t-test, and as we know, anything over 0.05, it has statistical difference, and that does have a couple of contenders, which is 714, 712, and 799, which indicate that there is a statistical difference because it's less than 0.05. However, again, it is important to note that these stocks were not observed at the same rate. The 324, 859, and 303 were observed. W1118 was the one that was observed the most because it was a stock present as a control. So it's quite important to understand that. Now we're going to move on to the female explanation of the data. It is laid out in the same manner as the male. We have the ST50 highlighted in bright green. If we zoom in to the upper left, we can see that the temperature was also kept at 22. The type, date, and time is also laid out on the second line, as well as the type flies and the amount of flies inside each vial. The y-axis shows the time as well, and if we move to the right, we can see the calculations. We did observe less female stocks, and we have 3 to 4 in W1118, which the error bars overlap, as well as 859. 714 does not. The y-axis represents the time, and the x-axis represents the stocks that we observed. We have statistical difference on only one of the stocks. It is the stock that we observed the least, so it is quite important to note that since we do not have enough observations, we cannot come to a conclusion. We're quite surprised to see the 714 has such a high, such a low tolerance to alcohol and became so inebriated so quickly, making it a perfect candidate to maybe make a study in parallel to the one from that was published in the Journal of Psychology. Now 3 to 4 does display about the same as W1118, but it does display a higher sensitivity to alcohol. That is important to note. In the male, we do have 714, 712, 799 displaying the highest sensitivity, but again, important to note that the ones that display a statistical difference, a significant difference, were also not observed enough. Note that although we did observe 3 to 4 more, we noticed that the following stocks, 714, 712, and 799, observed a natural inclination to becoming sedated. And the problem is that we could not finish all of our observations of these stocks, so more research is necessary. To prove statistical difference between the stocks, we used a t-test in which we compared the W1118 data to the stocks, and that's how we determined the t-test. It is important to note that the stocks displayed, not all of them show all of the observations and the rigor of the method, because again, COVID-19 did get in the way. It did not allow us to finish our research. Now we're going to take a look at the female data, and it is laid out in the same way that the male data is laid out, in which you have the x-axis with the stocks, and you have the y-axis being the time that it takes for the flies to succumb to ethanol sedation. It is actually important to note that although 3 to 4 did have a slightly that it was still not significant as compared to the W118. Now, something that was really different was to observe the stock 589, which actually display a higher tolerance than all of the other stocks combined, thus making it a really good candidate to further research. And most psychiatry set out on a similar path as the NCCU team. Their impetus was slightly different. The team set out to find SNPs which aided in the desire of consumption of alcohol and displayed higher sensitivity. In their search, they found potential stocks which show a natural desire to alcohol prior to being exposed, unlike the wild type, which look for alcohol once they have been inebriated. These particular stocks that most psychiatry searched showed this desire for alcohol consumption without being inebriated first, and that it is quite interesting. The genes that they looked for, these stocks that they looked for, were the team at Mold Psychiatry did set out their data a little bit different. Their y-axis represents the fly type, as the x-axis is the time that it takes for them to be sedated. It is actually inverted to the way that we set out our data. They actually looked for ARFIP, ARF5, and EIFIA as the stocks, and these were the ones that they looked for. These stocks, like I said before, showed a natural sensitivity to alcohol. They also showed a natural desire to consume alcohol. In conclusion, our research does not show a strain with higher resistance than that of the wild type, but we may have found candidates for further research. The flies that show higher sensitivity were DGRP 714, 712, 799, and this was in the female and in the male, while the 589 female stocks show a higher resistance than DGRP 324, which was, out of all the stocks, the one that was showing the higher resistance. As we know, higher resistance to alcohol inebriation may aid in alcohol use disorder, because as a person becomes more resistant, they need more alcohol in order to keep that buzz. leading them to increase their consumption, which then yields a even higher resistance. And this vicious cycle leads a person to consume a lot of alcohol, which is detrimental to the health. This is my observations and the data presentation that I have on the EMBA essays that we have concluded across the semester. If you have any questions, please let me know. Thank you very much. This is Jefferson Susoni. Have a wonderful day.